Gherard Batisti Biffignandi. I am a postdoc researcher in Hospital San Matteo in Pavia, northern Italy. And today I'm going to show you this poster that talks about a novel method to infer the emergence rate of transmissible traits in bacterial populations. So bacterial evolution is largely driven by horizontal gene transfer by mobile genetic elements such as plasmids, which can be either detrimental or advantageous for the host according with the exerting selective pressures. Once such kind of trait is acquired, for example, a plasmid carrying antimicrobial resistant genes, it can drive to selective events, and so enhancing the spread of these genes across and within bacterial populations, giving an evolutionary advantage. Despite the large prevalence of this trait in bacterial populations, their dynamics is not fully understood. And with the advent of genomic epidemiology and sequencing, we were able to provide a better understanding about their prevalence in the populations. But unfortunately, a lot of studies and methods are based on, merely rely on prevalence, which offer a limited insights about their evolutionary trajectories of these traits. Luckily, phylogenetic structure can be exploited to spot signals of convergent evolution and signatures of growth that may indicate an expansion in a subpopulations. So for this reason, here today, I'm going to talk about EMERGENE, that is a novel method which pairs ancestral state reconstruction with analysis of phyletic patterns of time-scaled phylogenetic trees in order to provide a proxy of and how much and how fast a gene is emerging through a population. Specifically, EMERGENE provides three main indexes. The first is the number of phyletic events that are actually the number of introduction of a gene across the tree. And then the entry rate, so basically the migration across demes that can be intended as a migration event, and the migration duration is calculated in order to understand how much time did it take the gene to be inherited from a parent node that was not carrying the trait to its child. And finally, the emergence rate, which is the most significant metrics that actually measure the evolutionary time that occur for a trait to be gained and then inherited through their descendants. We test the reliability of our method on both simulated and real case scenario. Talking about the simulated one, we simulated the evolutionary trajectory of 4 populations that were evolving at different pace, and the simulation was performed following a structured coalescent trajectory, which account for migration events, and we set that 3 populations out of 4 were experiencing different rate of clonal expansions. And we benchmark, as you can see here on the left, the prevalence. So basically we assume, we empirically assume that that each simulation harbor, all the individuals of each simulation harbor a specific trait. And under this assumption, we compare the standard prevalence, so basically what we would observe from the normal approach in epidemiology, compared with what we obtained from EMERGENE. And we correctly predicted that the population blue and purple, that were the ones that were set to having higher rates, actually were detected correctly. That exactly the inverse of what we would observe using the standard prevalence. So then we infer the number of introductions and also the migration rate. And we saw that actually the purple one that was set to have the lowest entry rate, we correctly predict also this one. And also we correctly predicted the number of introduction that we set in the simulations. So in case of the purple one, there was only 1 monophyletic events. And in case of the yellow and the blue, we set 2 phyletic events. And finally, we test our method also on a comprehensive data set of about 3,700 genomes retrieved from the UK SHA collection of Shigella sonnei pathogen. And here in this framework, we can see, starting from the top, the number of phyletic events for each gene that were predicted using AMR Founder Plus. And then we calculate the entry rate and the emergence rate, so all the three indexes. And representing like this output in this way, it's kind of like a GWAS approach, so like similar to a Manhattan plot. So basically, we spot that genes such as MPHA, that is related to azithromycin resistance, and the BLA C6M were having the highest emerging rates as expected. So this means that this method was correctly was reliable to predict this kind of spread. Also, the QRNS, that is responsible for the ciprofloxacin resistance, that was already shown in Shigella to have a previous spread over the last years. So to summarize, static prevalence are not always useful because, of course, they can be affected by some biases, for example, sampling, and also biases due to the clonal population, the clonality of bacterial population. And this method is fast. It works on time-scaled tree, so it can be used, for example, using backdating and beast tree as input. And then resistant genes or virulence genes or trait in general, categorical trait, can be tested within the same population to measure their different rates over time. And of course, most important, it can discover overlooked traits that were not observed to be responsible of recent spread. So basically, this kind of approach is also helpful in order to spot genes that are rapidly emerging but possibly overlooked due to their recent expansion. So thanks everyone for the attention, and please visit the poster in the next session, and a special thank to all of my collaborators.